Initially, the different arterioles carry a steady supply of blood in and out of the glomerulus, and here the filter is formed when the high blood pressure facilitated by random forces fluid out of the blood into the lumen of Bowman's capsule, which is not permeable to plasma proteins, red blood cells, and white blood cells. From here, the fluid is processed into the proximal tubule. As the filter progresses through the proximal tubule, hydrogen ions are actively transported and ammonia ions are passively transported into the tubule. So simultaneously, sodium chloride, amino acids, and glucose are actively transported, and water, potassium, and bicarbonate are passively transported into the interstitial fluid, then to the peritubular capillaries, which also rid themselves of processed drugs and toxins. Then the filter moves into the loop of Henle. As the filter moves into the descending loop, numerous water channels formed by aquaporin proteins make the transfer epithelium freely permeable to water, but not other salt and other solutes. This reduced solute permeability leads to the solute concentration of the filtrate to increase. After the filter rate reaches the tip of the loop, it begins to ascend. The ascending loop contains sodium potassium pumps, so no water channels. A hormone aldosterone increases the number of these pumps. A shift in permeability diminishes the filtrate solute concentration. The filter then reaches the distal tubule, where potassium ions and hydrogen ions are actively transported into the tubule. Simultaneously, sodium chloride and bicarbonate are actively transported, and water is passively transported into the interstitial fluid.
Lastly, the remaining filter is passed into the collecting duct, which regulates the concentration in the rear, while the ADH makes the collecting duct permeable to water, thus reabsorbs water into circulation by increasing number of water channels into collecting duct and increasing osmolarity of urine.